Meiosis 1, remember, is preceded by the S phase, so that means that the chromosomes and centrosomes are going to be duplicated. They're going to undergo prophase 1. They're also going to undergo prometaphase 1, metaphase 1, anaphase 1, and telophase 1. It is important to note that because the process of meiosis occurs in 2 rounds of division, that you have to refer to these phases as being part of meiosis 1 or being part of meiosis 2. So we are now going to consider what happens in the first round of division. In prophase 1, very similar occurrences happen, such as the chromosomes are condensing. However, in prophase 1 of meiosis, a very important event happens. This is called the crossing over event. This allows for the genetic exchange of material between the chromatids and also between the homologous chromosomes. This means that once crossing over occurs, we have genetically distinct chromatids. This does not happen in mitosis, and this happens specifically in meiosis because we have the formation of what is called a tetrad. Remember, we have our DNA being copied, so that means that we have our dad's original copy of chromosome 1, for instance, and we also have chromosome 1, our mom's original copy. Because these chromosomes contain similar genetic information, they are going to be held in close association with each other. This forms a tetrad. Tetrad refers to the fact that there are 4 chromatids in a tetrad. That means that our dad's original copy and mom's original copy for chromosome 1 forms a tetrad. We would also have a tetrad forming for chromosome 2, which means that for chromosome 2, my dad's original copy and mom's original copy are also going to be held in close association. And this will happen to all of the chromosomes within the genome. Once you have a tetrad forming, these chromatids will be able to cross over. This occurs during synapsis. This refers to the crossing over event. We can also call this homologous recombination because you're going to have the exchange of material between homologous chromosomes. This is very important to understand. It's also important to remember that we're dealing with chromosomes that contain the same genetic information. We have chromosome 1 from our mom and chromosome 1 from our dad that are forming a tetrad. This means that we're not losing any genetic information when we have the crossover event. We are simply exchanging genetic information. This is very important because if you have this occurring between chromosomes that aren't homologous, you will have mutations occurring. So it has to be between homologous-like chromosomes. The point of crossing over or attachment between these chromatids can be called the chiasma. You can have more than one chiasma occurring in a tetrad. So this can exchange a variety of genes between parental and maternal originating genes. This image here depicts that crossing over event. So in the crossing over event, because these chromatids are housed so close together, you can get the DNA twisting around and breaking off and exchanging at the crossover or at the chiasma. Once again, you have this occurring between paternal chromatids and maternal chromatids. Meiosis 1 will then proceed through the following phases. We have metaphase through telophase. However, because we're dealing with meiosis 1, we will refer to them as metaphase 1, anaphase 1, telophase 1. Metaphase 1 occurs very similarly to what we've seen in mitosis, where the genetic material is going to be lining up on the metaphase plate. However, because we are dealing with metaphase 1, we're going to be discussing the tetrads. We have the formation of tetrads in prophase 1. So in metaphase 1, these tetrads are going to be lining up on the metaphase plate. When we have anaphase 1, we are going to be separating out those tetrads, meaning we're going to separate out the homologous chromosomes. Eventually, this will then undergo telophase and cytokinesis, where we will produce 2 genetically distinct daughter cells. When we enter into meiosis 2, we enter into meiosis 2 immediately, which means that there is no genetic information that is going to be copied. There is no S phase, there is no additional growth phases. We simply complete meiosis 1 and enter directly into meiosis 2. This image here depicts meiosis 1. Note that in prophase 1, we have the formation of the tetrads and the crossing over events. This is very important and unique for the process of meiosis. This allows for the exchange of genetic material. We note that in this diagram we don't have prometaphase 1, but that would be essentially when the centrosomes are extending out their microtubules and those tetrads are being pushed towards the metaphase plate. When we enter into metaphase 1, we can see that all of these tetrads are lined up on the metaphase plate. In anaphase, note that now we are separating out those homologous chromosomes. Also note that on some of the chromosomes you can see different colors. So when the chromosome is primarily red, meaning that it originated from the maternal side, we can see that one of those chromatids actually has a portion of blue associated with it. This would have been from exchanging that genetic material with the paternal chromosomes during prophase 1. We can also see that on the primarily paternal chromosomes, the third chromosome at the top of the anaphase cell, we can also see that there are 2 points of crossing over that occurred in those chromatids. And we can see the exchange of genetic material such that the blue chromosomes have small little red tips. We then have telophase and cytokinesis dividing up and reforming the nucleus, dividing up the cytosol into the 2 daughter cells. At this point, remember, these daughter cells are considered to be haploid because they only contain one version of the gene. However, that version of the gene has 2 chromatids. In meiosis 2, we have a secondary round of division. And remember, we're not recopying out any DNA. So under meiosis 2, we have, once again, prophase, where the nuclear envelope is going to dissolve. The chromosomes are going to start to condense. You have the appearance of the asters. The microtubules are growing from the centrosome, and the centrosomes are starting to orient themselves to the opposite ends of the cell. You would also have prometaphase 2, which is skipped in this diagram, which would correspond to the microtubules latching on to the kinetochores and pushing the chromosomes towards the metaphase plate. Here in metaphase 2, we can see that we've got the chromatids aligned. So the simple chromosomes, either maternal origin or paternal origin, are lined up on the metaphase plate. In anaphase 2, we are going to separate those chromatids, and notice that these chromatids are not identical to each other. Why? Because we had the crossover events. So each chromatid is genetically unique. In anaphase, we're separating out those chromatids. The microtubules are shortening and pulling them towards the centrosome, and eventually this will undergo telophase 2, where we get the reappearance of the nuclear envelope, and we also get the appearance of the cleavage furrow. Eventually, this will form the 4 daughter cells that originated from the one original pre-gametic cell. During meiosis, there are many different reasons why the gametic cells or offspring are genetically unique and do not resemble the original parent cell. One of those reasons has to do with the law of independent assortment. During metaphase 1, when the homologous pairs of chromosomes, meaning that the maternal original in copy and paternal original in copy, pair up, they're oriented randomly as to which pole they face, which means that when they become separated out, it is going to be random which one, maternal or paternal, is pulled to either pole. This leads us to the rule of independent assortment. This means that for each tetrad that is formed, when this tetrad is separated during anaphase 1, it is separated out randomly, meaning that, let's say I have chromosome 1 and I have the tetrad forming. So I have my mom's original in copy and dad's original in copy forming a tetrad together. When this chromosome and when this tetrad separates out and those homologous chromosomes are separated, let's say, for instance, my dad gets pulled to the left of the cell and my mom gets pulled right. When I get to the next tetrad, that separation of homologous chromosomes is independent, meaning that it doesn't matter how the first tetrad was separated out. That has no influence on how chromosome 2 is separated. So just because my dad's went left and my mom's went right in chromosome 1, when I get to chromosome 2, that has no influence. So maybe in chromosome 2, I have a 50-50 chance of my dad going left and my mom going right, or vice versa. Similarly, when I get to chromosome 3, once again, it's a flip of a coin, 50-50 chance of my dad going left or right and mom going left or right. So that means that when we line up all of those chromosomes on the metaphase plate, they are sorting themselves completely independently of each other. And that is, in essence, the law of independent assortment. So this greatly increases the genetic potential that we have in producing the gametes. This gives us just an easy depiction of the law of independent assortment. So once again, let's consider that we have 2 chromosomes. Here we can see that the chromosomes have been duplicated, so I have my dad's original and copy in blue for chromosome 1 lining up and forming a tetrad with the mom's original and copy of chromosome 1. Similarly, I also have that for chromosome 2. When I have them undergoing meiosis, let's say that they separate such that my dad's chromosomes get pulled in the same potential gamete as well as my mom's get pulled apart together. So in essence, I have these gametic cells where half of them have my dad's genetic information and half of them have my mom's. But let's take a look at a different possibility. So now let's say that when these chromosomes line up and form tetrads, I have a different arrangement, such that my dad is on the left and my mom is on the right. And for chromosome 2, I have my mom on the left and my dad on the right. That means that when these tetrads get separated, they're going to separate out in a different arrangement, such that my daughter cells, you can see, have a completely different combination than what I had in possibility 1. Consider that we have 23 different chromosomes for this to occur in. So you can start to see the different combinations that we can have can increase the variability astronomically. So for 23 chromosome pairs, there's a unique combination of both maternal and paternal chromosomes for each gamete that we produce. Another source of genetic variation has to do with the fact that we have random fertilization of gametes. That means that it is random with respect to the genetic combination that will end up fertilizing the egg. That leads to many different possible zygotes that we can have. Now, fertilization isn't completely random, meaning that there is some evidence that there's different sperm characteristics that will allow for an increased probability of fertilization. Another source of variation has to do with that crossover event that we examined that occurs during prophase 1. You can have multiple crossover events happening along a chromosome pair. So this can greatly increase the exchange of genetic material. This can reshuffle the deck of paternal and maternal chromosomes and increase the variability of the genetic gametes that you can produce. So that means there's an infinite combination of genes that could end up joining together to undergo syngamy to form a zygote. This creates a considerable amount of variation in the population. And remember, this is very important so that natural selection can work.